Hello and welcome to the poster. So in my lab, we investigate how the primate visual system encodes information about the world. And we think that the key problem to look at is the idea that the world has a lot of information in it, but only some of it is useful to a given organism that might care more about patterns present in faces or body parts than those occurring randomly in the background. So our overarching theory is that neurons in the primate visual system are learning to encode specific combinations of visual patterns that are diagnostic of food, friends, or foes. So the questions in this poster are, what are these patterns? How complex are they compared to the natural world? And how do they relate to the natural world per the organism? Okay, so the first question is, what are these visual patterns? And to extract them, we use a neuron-guided closed-loop approach that we published in 2019 that relies on a deep generative network, or what we call here a generator, which can create random samples like these kinds of textures. When coupled with an evolutionary algorithm, this system allows a neuron to guide the development of images in such a way that they become highly activating. And the advantage of this approach is that in compared to images used in the past that are a little oversimplified, they're much harder to relate to the natural world and don't really activate neurons as well as natural scenes do. The images that we can extract from this particular approach are activating and can perform just as well as natural images, and they are easier to relate. So the first question is, what are these visual patterns? And we've run hundreds of experiments across the ventral stream of the macaque. And here's some examples. Here's 6 different experiments involving 2 particular sites in inferotemporal cortex. And as you can see, the patterns can be as simple as a yellow contour that could be used for diagnosis of food, or more complicated patterns that could be useful for the detection of faces. So across all of these experiments, we noticed 3 major observations. The first one is that these prototypes do contain patterns that are present in images of real-world objects, so in images that are also highly activating to the neurons. Prototypes are not full scenes or objects. They seem simpler, and they seem to be clearly segmented from the background compared to random generator samples like you can see here. So that leads us to the second question of how complex are these prototypes compared to natural objects and other kinds of references. And to answer that question, James Johnson, a postdoc in the lab, measured the compressibility of the prototypes using a simple discrete cosine transform, which is kind of like a Fourier transform. And it's a little bit involved, but suffice it to say that this ratio has low values when trying to describe an image containing an object that is well constrained or a specific object part, and it's a large value when it's describing an image containing an extended irregular texture. We found that prototypes tended to have a complexity that was lower compared to random generator images produced by the exact same model without neuronal guidance. They were also simpler than the kinds of representations present in classically trained neural networks like AlexNet, and simpler than random images from the natural world. Interestingly, though, they were also more complicated than the classic stimuli that have been used in previous visual neuroscience studies used to characterize the function of ventral stream neurons. So our main conclusion is that the ventral stream neurons are encoding a density of information that is comparable to object parts. So that leads us to the final question of how do prototypes relate to the natural visual world? And here, our grad student Olivia took the approach of thinking that when monkeys are allowed to look at natural scenes, they tend to choose on salient features like faces or body parts. Prototypes often tend to resemble things, patterns present in faces and body parts. So perhaps if we let the animal just free view thousands of images, it would give us an idea of regions in the natural world that do contain the same prototype. So we did that experiment. We presented thousands of images to different monkeys, and we cropped every location where they actually fixated. And we propagated these image fragments into AlexNet along with prototypes that were recorded from the brain, samples from the generator that without neuronal guidance, as well as image fragments coming from the same natural scenes that were not viewed by the monkeys. And all of this was in order to visualize the different distances between these groups. And what we find is, first of all, there are 2 major clusters. The first cluster corresponds to images from the generator. The second cluster corresponds to images from the natural world. And as you can see, they are different because the generator does not create photorealistic images, but even within these clusters, we can find that the prototypes are closer to the fragments of the visual world that the animal also viewed, suggesting that these ventral stream neurons really are encoding visual patterns in regions where the animal looked more frequently. So that's it. This work has been accepted in principle and intercommunications. It's under editorial revision. If you have any questions, please let us know. This is our website. And thank you to our funding sources and you for your attention.